Remember all the subatomic particles, the electrons, the protons, and the neutrons, where they're located at. Do I? In the cookie.
Gotta have a Pop-Tart, man.
Gotta have a Pop-Tart.
My breakfast.
Remember, the protons and neutrons are in the nucleus, and the electrons are in
The balance.
shells, or they orbit around, right? Now remember, protons have a positive charge, neutrons a neutral charge, electrons a negative charge. Some things that you've already learned previously too, the atomic number is the number of protons. And keep in mind that before the atom, if for some reason it doesn't have those outer shells complete, you guys remember the first three shells, the numbers to those?
2, 4, and
2,
2,
8, and 8.
8, and 8.
2, 8, and 8. So remember, if it's not complete, those electrons will either be transferred or gained, or they'll share electrons. So before that atom is entered into any bonds to make that outer shell happy, remember that the protons and the electrons are equal. So as you see it on the periodic chart, before it forms any bonds, those protons and electrons are equal. Now the atomic mass number, that is the number of protons and neutrons. If you actually measure the atomic mass in AMUs, that's all the subatomic particles at rest. Electrons contribute very little to mass, so it only goes out a couple of decimal points. They're really the same number, but the atomic mass technically has that decimal point when it's got the term or word number after it. It's just a nice rounded number. You don't have to worry about atomic weight. Nice picture of the atom. An element you guys remember that is a pure substance. Only one atom in an element, so you do not find hydrogen and oxygen and so forth. You don't memorize this table by any means. I just want to bring up another concept if you want to write to the side in your notes. If you take a look, carbon, hydrogen, they all have some isotopes that are listed. And you guys remember what changes in the isotope. Now the atomic number is still the same. So protons, electrons, those are still the same. So the thing that changes in the isotope is the number of neutrons. Now that'll change the atomic mass or the atomic weight. So the isotope, what changes is the number of neutrons. The atomic number remains the same, but the mass number or atomic weight, those are used at different times, but they would change due to the change in the neutrons. So atomic mass or weight would change. Don't memorize any of these. Cobalt, they've got a cobalt and iodine. Not anything you have to do. I just want you to know what changes with that isotope and what remains the same. Difference between molecules and compounds. Now, a molecule is just the combination of two or more elements. It can be the same or it can be different elements. Not really any specification. They are held together by chemical bonds, and specifically the covalent type of bond. Now a compound, you have to have the combination of two or more different elements. So I've got water as an example for a compound, and any of the diatomics fit, but a hydrogen up there for the molecule. H2, O2, N2, any of those little guys work. But a compound can have either a covalent bond or an ionic bond. So what happens is that you can have a molecular compound or an ionic compound. So that example H2O, if you guys remember, it is covalently bound. So it is also a molecule and a compound. Then now take sodium chloride. It has an ionic bond. That only fits into the compound category. I know we went through this in A&P, but you guys remember all this? So the molecule doesn't necessarily have to be different elements, but the compound does. But the molecule has to be covalently bound, whereas the compound can be covalently or ionically bound together. So H2O, since it has a covalent bond in between the H's and the O's, or O, that would be a molecule and also a compound. But remember, sodium chloride, no covalent bonding, so it only fits into the compound category. Any questions?
Sodium chloride, no.
Sodium chloride, only a compound because it's an ionic bond. And just think about it, when you see the Na pluses and the CO minuses, the Na plus, that means it's lost electrons. CO minus means it's gained. So if any of those, if you see any of those ions, then you know that they're going to be attracted together by that positive and negative charge. So that would be an ionic bond. So ions will be attracted together by the positive, negative charge. So anything that would be an ion that would form that type of bond just goes in that compound category. Now, sometimes you'll see like 2 plus, 2 minus, that just tells you how many electrons they've lost or how many electrons they've gained. Keep in mind when they've got the pluses, their protons now outnumber their electrons. When they got the minuses, the electrons now outnumber their protons. So that's why you got the different charge. So just reviewing again through those chemical bonds, remember covalent bonds, they share electrons. And they share electrons in that effort to complete that outer shell. Now you can have more than 3 shells, but typically everything that we study, we just go up to the third one. Now ionic bonds, remember there is a transfer of electrons. Someone loses, someone gains. And we already talked about that. Those are typically hydrophilic in nature, which means they'll interact with water. Now covalent bonds, remember what polarity is. You can have polar and non-polar covalent bonding. Polarity is just referring to the charge distribution. So, how those electrons are distributed, if it's evenly along the molecule, then it'll be nonpolar. If you have more electrons fall to one end than the other, it will be polar. And that will result in a positive pole and a negative pole. So water is a great example of a polar covalent bond. Just look at this with me, you don't have to go all the way over here. If you take a look, the O, towards the O, you got 1, 2, 3, 4, 5, 6 electrons. Everybody sees that? Towards the H's, 1, 2, 3, 4. So that means that the O would be more negative than the H's. So one side is more negative, one side is more positive than the other. So that creates an unequal distribution of electrons, which creates a positive and negative pole. Things that are nonpolar, they're equally distributed along the whole thing, and there's no different poles to it. And if you think about things that are a lipid base, they would be nonpolar. They don't interact with water. So things that things that are hydrophilic will have a charge, and you can be attracted to one side, either the negative or the positive pole. So let's go back to that hydrophobic, hydrophilic. Ionic bonds are hydrophilic in nature. But now, when we just talk about hydrophilic and hydrophobic, remember that term hydro, we're talking about the relationship to water. So if something is hydrophilic, likes water. If something is phobic, it's scared of water. So things that are polar are hydrophilic. Things that are nonpolar are hydrophobic. So oils have equal charge distribution, and they don't want to be anywhere near water. So if you drop oil into a glass of water, it tries to stay together as much as it can. Now the last type of bond, if you guys remember, hydrogen bonds, those are the weakest bonds, very important in things like nucleic acid structure, also protein structure and so forth. Hydrogen bonds are located between water molecules. Now this definition's a little long. It's formed between hydrogen covalently bound to a molecule and an oxygen or nitrogen atom on the same or different molecule. That's a lot. An easier definition is it just results from polar covalent bond. So if you got a bunch of polar covalent bonds, you're going to find some hydrogen bonds. Hydrogen bonds are easily broken apart. Covalent bonds are the strongest. Ionic bonds, given it the right situation, will break apart really easily too. So ionic bonds and hydrogen bonds are a lot less stable than that covalent bond. All of these are just pictures. Now, take a look at water here. Now, let's say we were to drop sodium chloride in here. As that sodium chloride dissolves in water, where will the Na pluses go?
Towards the negative ones.
Right, towards the oxygen end. Where will the Cl minus go?
Towards the plus.
Towards the hydrogen end. And there's a picture in your, I think this picture's still in this version of the book, that shows which would be where.
Bless you.
And this is, bless you, what makes water such a good solvent is because it has those poles. So it can easily break up these ionic bonds.
Yes.
Now, if you take a look at this picture, you can see in like that, it's like a seafoam green or something, where they have the water molecule. And within that, they show the straight lines, and that's indicating stronger bonding. So this right here is indicating the covalent bond, where they show you the dashed lines, that's saying it's a weaker bond. That's your hydrogen bonds. So you find the covalent bond in between the H's and the O. You find the hydrogen bond in between water molecule to water molecule. Is everybody following along with me?
No. I'm looking at this picture and it's not making sense.
Okay. So this whole thing right here, these outlines here, that's just water.
Okay.
This is indicating that it is a covalent bond between the H's and the O. So from water molecule to water molecule, you have a hydrogen bond in between each.
Okay.
So now as things dissolve, these will break apart as something will go towards one end, they can be reformed. They're easily broken and reformed quite a bit. Even in proteins, it works that way as they change shape as
Okay.
well. So the important properties of water, I would like you to know these: capacity to form hydrogen bonds with other water molecules, and that's indicated by the dashes. It is polar, so it makes it a solvent. So things that are hydrophilic will dissolve, things that are hydrophobic will not dissolve. So I've given you that oil as an example of something that is hydrophobic. Now, if you guys remember, the cell membrane is organized with hydrophilic parts and hydrophobic portions, and it's a phospholipid bilayer. And that's how that cell membrane is put together. And we'll talk about this again with the, when I get to the phospholipid slide. But remember, those nonpolar only face each other, and the hydrophilic face towards the water inside or outside of the cell. All right, that's a lot of the basics. Now, pH. Don't memorize the pHs of all this stuff. I'm not really interested in you doing that. The pH scale, remember, is a measurement of the hydrogen ion concentration. And H plus means hydrogen ion. OH minus means what? What is that? More basic. That's hydroxide ions, right, and it is more basic. So H plus, and you'll see it written out that way on the exam, H plus is the hydrogen ion. OH minus is the hydroxide ion. Now, I usually do a test question on this, so make sure that you know these rules here. If you have a solution that's neutral, you have equal amounts of hydrogen and hydroxide ions. You have a solution that's neutral. The hydrogen, the H pluses, are the same as the OH minuses. Now, if something is acidic, you have more hydrogen ions. If a solution is more basic, you have more hydroxide ions. And you guys realize, I mean, 6.9, that's still slightly acidic, so anything below 7, and anything above 7 is going to be alkaline, even if it's 7.1. Nice, easy definition of organic versus inorganic. So as far as these different types of molecules, if something is inorganic, you will have carbon or hydrogen present. So just because it has carbon doesn't mean that it is organic. Carbon dioxide or carbon monoxide, those are both inorganic. But to be considered organic, you must have both carbon and hydrogen present. You guys don't have to memorize any of those. We can go on to the macromolecules. Not really an organic chemistry class. Try to keep it as close to the A&P as possible. Now you got to know your 4 types of macromolecules. Remember, that's carbs, proteins, lipids, and nucleic acids. Starting out with the first group, carbohydrates. Tell me the difference between the monosaccharide, the disaccharide, and the polysaccharide.
1, 2, and multiple.
1, 2, multiple. Yeah. Now, all of these examples here have 6 carbons, and we've talked about this before in A&P. When we get into the nucleotides, they're made up of pentose sugars that have 5 carbons, but all these have 6. So the monosaccharide, right, is 1 6-carbon sugar. The di is 2 6 carbons put together, and the poly is as many as you want. Now, some examples here that I have on, for monosaccharides, I want you to know glucose, galactose, and fructose. And I will not have many of these pictures, but they are nice. They show you all the carbon positions. And as they're labeling 1, 2, 3, 4, 5, 6, that's telling you where the 6 carbons are located at. Usually they're points, but now sometimes they'll have some that kind of hang up a little bit. Some examples here of disaccharides: maltose, sucrose, lactose is also. Now, one of the things that I want you to know is very important on this slide. Remember, as you are putting these monosaccharides together to make a di or to make a poly, you're building up, so that process is anabolic. If you were to break it apart, that process would be catabolic. So when these sugars are put together, they're put together with dehydration synthesis, and water is formed at the end of that process. Now, hydrolysis is catabolic because you're breaking apart with water. So when I'm putting these monosaccharides together to make a larger disaccharide or a poly, dehydration synthesis is used. So that's building up, that is anabolic. Now the bonds that form between these sugars are called glycosidic bonds. And I will ask you that on the test as a description. So whether it's a disaccharide or a polysaccharide, they're put together with glycosidic bonds. And when that bond is formed, that's dehydration synthesis. You guys remember all that? So anytime you're building up, that's dehydration synthesis. Anytime you're breaking apart, that's hydrolysis.
And when you're building up, you're using the water molecule to build it,
You're taking it out, so water's removed with dehydration. Dehydration.
Okay.
With hydrolysis, you're using it to break it apart. And if you take a look at this picture, you can see where the water is taken out to make maltose. So the 2 H's and the O's where they come from to make maltose, and then that gives you water along with it. Now, if you were to do hydrolysis, it'd be the reverse. So you'd break apart with water, and the H's and the O would stick back where they were. So you put water back where it originally was, so to say. So dehydration synthesis, water's removed. Hydrolysis, water breaks apart.
Okay, I'll be the one to ask all the questions. Does anybody struggle with these diagrams besides me, or are you all able to look at that and follow exactly everything? Okay, I can honestly tell you as I go through the book and read, I get to some of
You gotta
these.
look at the highlight part. So this is where your water's coming from.
Okay,
And once that water's removed, that bond is formed between those two, but you end up with a water molecule.
so all they're
So
showing me on that
that's
second row
dehydration.
is the end result is the water.
Right. And this is a different sugar. They're putting 2 different sugars together, but same thing.
Okay,
They're removing water, and you end up with sucrose in that instance and the water.
okay,
They didn't show you the reverse, but it would just be taking water plus maltose and putting the H's back and the O where it belongs.
okay.
That would be hydrolysis. The A&P book had a really good, and I can't remember, I think it was in chapter 4, you guys were under the holes, in chapter 4 where they showed you the reverse. So I don't know if you wanna look that up at all, but Now cellulose and starch, those are good examples of polysaccharides, but now this is a weird one that doesn't quite follow all the rules that you want it to. Peptidoglycan, the cell wall of bacteria. It's a good example of a polysaccharide, but it's part protein and part carbohydrate. So just a little different structure.
Can you say that again?
Yeah, the peptidoglycan, still repeating sugars, but it's part protein that links it together. So the peptidoglycan is a little different from the ones that we just looked at in that it is going to be a bond that is going to be amino acids as opposed to carbohydrate, glycosidic. So peptidoglycan is a good example pertaining to micropolysaccharide, but it is part protein. That's what the peptido is. And I will actually teach you that exact structure when we get to Excel. So as far as this test goes, just know that's an example of a polysaccharide. All right, any questions about the sugars? We good there. These, every once in a while, the book too will have animations on the website, which is pretty decent. Not every chapter has them though. I think that the A&P book, if you guys kept that, was a nice basic introduction to some of those terms like the dehydration, synthesis, and hydrolysis. And I think it gave you examples for amino acids being put together, the sugars being put together. So it was an All right. You guys want a break or you want me to continue? I can probably finish with this whole thing. We can be done pretty early. If you guys need a break though, I'll let you because this is
It's warm in here.
I thought it was just me, but I am cooking.
We were back
I know
there.
it is very warm.
Huh? I'm not, it's hot.
We can open
Usually
that
it's
door if
cold.
you want to. I didn't think it'd get that hot in here. It's about like a sauna.
In here, usually we're like,
Freezing.
yeah.
Last term, we were freezing.
Very cold on the other side of the building, but yeah, it's a little warm. You guys wanna open that door, walk around for about 5 minutes or something and come back?
Yeah, I get that.